talking about a type of cell division known as meiosis. Meiosis is a cell division that occurs in the gonads and is responsible for creating sex cells, so that would be sperm cells in a male or an egg cell in a female. What we're going to do with this video is take you through some of the stages and talk about some of the important terms, the important vocabulary words and processes that lead to the formation of 4 cells with half the number of chromosomes. So let's go ahead and do that. Meiosis, again, is a type of cell division that creates sex cells. Again, sperm cells in a male and an egg cell in a female. So we're going to have to reduce that chromosome number by half. Humans have 46 chromosomes in their cells. So meiosis will create these sex cells with 23 in each. And that's important because when the cells unite in a process known as fertilization, we're going to restore the chromosome number. So the 23 in the sperm cell plus the 23 from the egg cell bring us back to 46. And that's the normal number for humans. And that baby would then grow, that zygote would grow into a baby. What we want to look at on this next slide is that the original cell winds up dividing 2 times in order to create cells, 4 cells, with half the number of chromosomes. So the cell replicates its DNA once, but it's going to divide twice. And this is also called a reduction division because it is reducing or lessening the chromosome number by half. Okay, so what we see on this slide here is a comparison between mitosis and meiosis. On the left here in tan, we can see this is mitosis. And we can see that we're getting 2 new cells, we're getting 1 division. The chromosome number is the same. If you count here, 1, 2, 3, 4, 5, 6, we've got 6 chromosomes up here, and we wind up with 2 new cells with 6 in each. That's mitosis. But on the other side here, this is what we're going to be talking about today. This is meiosis. And we start off with the 6, but what we're going to do is replicate that DNA. We're going to split it 1 time. So this is our first division. Here's our second division. So we're going to get 4 cells. And if you count the chromosomes in each one, we're going to get 3 in each cell. We've reduced the chromosome number by half. Those are going to be sex cells. If those were sperm cells with 3 chromosomes in each, they're going to unite with an egg cell with 3 chromosomes in each. It's going to restore that chromosome number back to 6. Let's go to the next slide. Now, when we deal with meiosis, we're going to have to talk about homologous chromosomes. They're going to be used in meiosis. They weren't mentioned in the mitosis video because they didn't really associate with one another. The DNA just replicated and simply divided. With meiosis, we have to pay attention to homologous chromosomes. Homologous chromosomes are chromosomes with traits found at the same location from each parent. So if you look here on the left, we have a chromosome from mom, and on the right, one from dad. These are homologous because you can see there's a trait here at the top and a similar one from dad. This might be mom's hair color and then dad's hair color. What gets expressed in the baby is due to genetics, but we have the same type of gene at the same location. What gets expressed might be a little bit different. Here, this might be maybe their blood type, mom's blood type, dad's blood type. These are homologous chromosomes. They're like pairs of socks. And meiosis involves the pairing up of these homologous chromosomes. When cells have homologous chromosomes present, we say that they are diploid cells. Diploid cells are the cells with the normal number of chromosomes. So for a human, again, that's 46 chromosomes. But if we have 46 chromosomes, that means we have 23 pair. 23 from mom, 23 from dad. We call cells with homologous chromosomes diploid, and we represent a diploid cell with this 2N, which you see down here. The goal of meiosis is to cut that in half, so they would produce 4 cells that are 1N, or we don't write the 1, we just write N. So these would be half the number of chromosomes, and those cells are called haploid or monoploid. They do not have the homologous chromosomes present, and that's really how we are going to define them, and that's going to help us later on as we look through the two divisions. When does the cell become haploid? Okay, so what we're going to do here is go through some of the stages of meiosis and introduce some really important vocabulary words. So the way that I'm going to do this is I have drawn out a bunch of cells that show a series of stages of division. And you can see, if you look right here, this is going to be our first division, and over here is going to be our second division. Okay, I do skip a couple of steps because I think you guys know about things like prophase, metaphase, anaphase, telophase. So some of them I skip just so that we could do the accounting of the chromosomes and what happens. Let's take a look at this top original cell. I've gone ahead and drawn some chromosomes in here. If you count them, you can see pretty clearly there are 4 chromosomes in here. I did color them a certain way. Red is from mom, blue is from dad. So you can see we can find homologous chromosomes here. The large red from mom is homologous to the large blue from dad, and the small red from mom is homologous to the small blue from dad. So we have 4 chromosomes, and we have 2 pair. And I'm keeping it with a low chromosome number just so that we can do the accounting pretty easily. The first thing that's going to happen is the cell is, through interphase, going to go through the G1. It's going to grow. It's going to go through S, so it's going to replicate the DNA, which you see happening in that second stage here. We make a copy of the DNA, and then it's going to grow a little bit more and get ready to divide, go through the mitosis stages. The DNA at this point is in a really thin form. It's called chromatin. It needs to shorten and fatten, and that's what you see in the next cell. Now, the thing that's different with meiosis is that these homologous chromosomes pay attention to one another. The large red that you see here is homologous with the large blue. And they're going to want to join. They're going to come together. And so will the small blue and the small red. They are going to join in this pro-metaphase and line up in the middle at metaphase. The name of the joining is called synapsis. Okay, so I'll write that in there. Synapsis is the formation or joining of these homologous chromosomes, and when they join, they form these structures here, and this structure is called a tetrad. Okay, this is a tetrad. So we see 2 tetrads. Synapsis forms them. Now, here they are in the middle of the cell. That's metaphase. In anaphase 1, they're going to break apart. But you might be wondering, they were already apart in prophase. Why would they line up in the middle for metaphase, only to break apart again in anaphase? There's a reason. When they form these tetrads, it allows an opportunity for one homologous chromosome to switch pieces or shuffle some genes with the other homologous chromosome. So it's called crossing over, and it looks like this. Now, where that happens is at a site called the chiasmata. Okay, so there's the chiasmata, and that's the location where the homologous chromosomes twist around one another and allow for exchange. Now, I'm only showing it happen in one location. That can happen all along where they meet, so you get this massive amount of gene shuffling. And that's going to be important because now they're going to separate. We can see that the tetrad is starting to break up, and when it breaks up, that's called disjunction. So disjunction is when the tetrad breaks up. Okay, disjunction. And then we're going to go through telophase, and we're going to get 2 new cells, and you can see that the ones here on the top and bottom have separated into this cell, and the one on the right has moved over into this cell. Now, remember our definition of diploid and haploid. A diploid cell is one that contains homologous chromosomes, or the normal number of chromosomes. This cell over here that we started with had 4 chromosomes and had homologous chromosomes present. So it was diploid. This was 2N. But when we get to this first division, are homologous chromosomes present anymore? No, they're not. So this cell has become N or haploid, and this has become N or haploid. We have to divide one more time in our second division, because what we need to do is we need to separate these sister chromatids. So they will line up in the middle. That's metaphase 2. In anaphase 2, they're going to break apart. And then you're going to get telophase 2, and these will move over into the cells. Like you see there, it's going to pinch in with a cleavage furrow. And you will get 4 new cells made in that second division. 4 new cells made. And if you do the accounting, remember we started with 4. Now we have 2 chromosomes here, 2, 2, 2. We've reduced that chromosome number in half. But if you look really closely, if you pay attention, look at the design of the chromosomes. We started with 1 large red, 1 large blue, 1 small red, 1 small blue. But if you look at in here, we've got some shuffling here. We've got a red with a blue. We've got a blue with a red. So each one of these sex cells is genetically different. And that's really important because it creates diversity in the sexually reproducing creatures. And each one of these sex cells, every time sex cells are made, you get a new shuffling, so you get a new combination, and that's why creatures that sexually reproduce are so diverse. One of the main things that I wanted to focus on with you guys is not only the important vocabulary words like synapsis, tetrad, disjunction, chiasmata, it was also understanding when does the cell become haploid. So if you look at this diagram here, we start off with our original cell. You can see that it's 2N. It goes through the first division, meiosis 1, and we're already haploid. Why do we need meiosis 2? Because here you've separated the homologous chromosomes. So you had this situation where you had the homologous chromosomes present as a tetrad. Now you separate them here, and you need that second division to get each piece alone. So in the second division, you're separating sister chromatids. In the first division, you're separating homologous chromosomes. That's the difference. You wind up with haploid cells, and those are sex cells. Okay? So I hope that helped, and I will see you next time.